All right, how's it going, everybody? Welcome back to Engineering Major, Philosophy Minor. I hope you guys are having a great Saturday evening. I actually have another business trip coming up to North Dakota tomorrow, flying out relatively early, so at least in terms of California time, 8:37 p.m. I'm gonna have to get to bed pretty early, but I did want to make a quick video before I got into bed this evening. And today we're going to talk about the COVID-19 vaccine efficacy data. There's a little article up on here that I thought was kind of interesting, which is basically just led me on a path to doing some of my own research, and I came across pretty interesting information about the Pfizer vaccine. So before we get into that, just really quick, want to do my little intro. EMPM's, of course, the spot where you can get a working aerospace engineer's opinions on topics pertaining to engineering, philosophy, politics, and various other social issues. I was watching some Tim Pool earlier today, and hopefully you guys watched Tim Pool. I think Tim Pool is a really awesome YouTuber. He's got a lot of great information. He does a lot of good research. He definitely does a lot better research than I do. But I saw in one of his videos that he put out earlier today this article about Australia having an issue with their COVID-19 vaccine because I guess some of the volunteers for the clinical trials had false positive HIV tests. After having the vaccine administered to them. Actually, it wasn't Tim Pool, it was Anthony Brian Logan, another good YouTuber. But anyway, that's kind of beside the point. So I did want to kind of like look at this article a little bit. I thought this was pretty interesting. And we'll just go and look at some of the key facts here. So it says, while there is no chance that the vaccine could cause an HIV infection, something detailed follow-up tests confirmed. Scientists warned participants that the vaccine could possibly generate a partial immune response to a protein fragment used to stabilize the vaccine, which is also found in HIV. So I am somebody who is very personally concerned about my own sexual health. Health generally, obviously, but just sexual health generally. So I definitely, if I had participated in this trial, I would have been freaking out, especially because they had to do follow-up tests in order to confirm that the vaccine couldn't cause an HIV infection. This is pretty sketchy. Anyway, when I read this article, I was like, I wonder if the type of vaccine that America has developed, like the Pfizer vaccine, the Moderna vaccine, and the AstraZeneca vaccine, I wonder if any of those, have use these proteins or something like that. Fortunately, it turns out that they don't. And in the process of trying to research that, I actually came across this really nice, comprehensive FDA briefing document. This was written by Pfizer and BioNtech, or I don't know if that's spelled BioNTech or pronounced BioNTech. But this document is actually really nice. It's like 53 pages long. It's titled Vaccines and Related Biological Products Advisory Committee Meeting, December 10th, 2020. So this was documentation that was provided to the FDA. It seems like almost really justifies like their efficacy number of 95%, which came out a few weeks ago. And then also their kind of like safety track record for the vaccine itself. So I won't read too much from this document, and admittedly, I didn't read the entire thing, but there's some pretty interesting findings in here. So the reason I actually wanted to make this video is because I know that there's a lot of people out there who are relatively skeptical of the vaccine. They're not really certain about the methods maybe used to develop it. They're concerned that it was done under a rapid timeline, and therefore maybe some corners may have been cut. In general, I wanted to show you guys this document, not because I'm trying to convince anybody that they should get vaccinated or that they need to get, they absolutely have to get vaccinated. We'll talk a little bit about that at the end of the video, whether or not I think it should be mandatory to get vaccinated. But I did want to provide you guys this document because maybe a lot of people haven't read through it. And I know that we've had, for example, Governor Andrew Cuomo, he says, well, 50% of people don't trust it, and he wants to make his own separate committee in order to like not only review these documents, but also what the FDA has approved. And, people are concerned simply because it was developed under the Trump administration, which I think is a little bit ridiculous. But if you want, I'm going to show you guys some of this document, but if you want, you can look up this document for yourself. You can read through it. It's very comprehensive. It gives a lot of insight into the types of methods that they use to test for safety and for efficacy. And we'll talk a little bit about a question that I had to do a little bit more digging on. But if you guys want to do your own research, you can definitely read through this document and kind of convince yourself whether or not you think that these methods are sound. And you don't have to go and follow the experts or listen to the media or blindly trust your politicians. You can actually go through and read this document, and of course I will share some of it with you in this video. All right, guys, so we won't go through the whole thing, but I do think that this executive summary right here, just like this one paragraph in here, does just provide a lot of really interesting information. Then we'll look at more elements in here as well. The EUA, that's the emergency use authorization. So that is the order that that Trump either utilized or signed or put into practice or whatever. That's what EUA stands for. In order to get this stuff done faster. It says the EUA request includes safety and efficacy data from an ongoing phase 3 randomized double-blinded and placebo-controlled trial of BNT162b2, that is the vaccine, the Pfizer vaccine, in approximately 44,000 participants. So right off the bat, you can see that their sample size is quite large. Now, the question would be, is that large relative to other vaccines? I'm sure that's maybe like a relatively normal sample size, but still, you can imagine that's quite a bit of people. So let's go ahead and continue. The primary efficacy endpoint is the incidence of COVID-19 among participants without evidence of SARS-CoV-2 infection before or during the 2-dose vaccination regimen. In a mid-November analysis of 36,621 participants randomized 1-to-1 to vaccine or placebo who were included in the per-protocol efficacy analysis population of participants without evidence of SARS-CoV-2 infection prior to 7 days after completion of the vaccination regimen, efficacy in preventing confirmed COVID-19 occurring at least 7 days after the second dose of vaccination was 95%, with 8 COVID-19 cases in the vaccine group and 162 COVID-19 cases in the placebo group. So that is a very, very long-winded sentence. Basically, what they have outlined here is they've outlined what the kind of like endpoints are for their their trial study. What kind of time frame that they're going to use in order to see if there is a COVID-19 response in both the vaccinated and unvaccinated groups. And as you can see, they only observed 8 COVID-19 cases in the vaccine group after this trial period and 162 in the placebo group. That's the group that did not receive the vaccination. That's already somewhat promising, but we'll actually go in and look at how they do like the risk calculation, how they arrive at this 95% number. So in addition to evaluating efficacy, so there's 2 different things that they're measuring right here. They're measuring efficacy, so that's the ability of the vaccine to prevent illness or infection with COVID-19. And there's 2 parts to efficacy. There's preventing illness entirely and or lessening the severity of the illness. So there's 2 things there. But then there's also safety. Vaccine safety is something that Andrew Cuomo, for example, has questioned a little bit and claims that a lot of citizens of New York State, at least, question. There have been polls about people questioning that say that a large percentage of people, both on both sides of the aisle, actually kind of question the safety of the vaccine. And so this document actually presents what Pfizer and BioNtech or BioNtech, however you want to say it, what they have compiled and what experiments they have done in order to verify safety, and it's actually very comprehensive. So I'll quickly just read this one last sentence from just kind of like the executive summary. Safety data from approximately 38,000 participants greater than 16 years of age randomized 1-to-1 to vaccine or placebo with a median of 2 months of follow-up after the second dose suggest a favorable safety profile with no specific safety concerns identified that would preclude issuance of the emergency use authorization. Available safety data from all participants enrolled through the November 14th, 2020 data cutoff, N equals 43,252, which includes, I won't read this parentheses stuff. November 14th, 2020 data cutoff was consistent with the safety profile for the approximately 38,000 participants with median follow-up of 2 months and also did not receive specific safety concerns. The most common solicited adverse reactions were injection site reactions, 84.1, so that's like your arm getting sore after you get a vaccine. Fatigue, 62.9%, headache, 55%, muscle pain, 38%, chills, 31.9%, joint pain, 23.6%, fever, only 14%, and then severe adverse reactions occurred in 0% to 4.6% of participants and were more frequent after dose 2 than dose 1, generally less frequent in participants age 55 and older as compared to younger participants. Now that's a good thing because if you're older and you have a severe reaction to the vaccine, it's more likely to be serious. The frequency of serious adverse events was low, less than 0.5%, without meaningful imbalances between study arms. And then this next sentence is interesting. I'm going to skip to this second clause here. There was a numerical imbalance of 4 cases of Bell's palsy in the vaccine group compared with no cases in the placebo group, though the 4 cases in the vaccine group do not represent a frequency above that expected in the general population. So what that basically means is that the rate of Bell's, if you were to calculate kind of the risk rate of Bell's palsy in the vaccine group, you would not find that it was significantly higher as a result of the vaccination by comparison to either other vaccinations or just events in the general population from other things that cause Bell's palsy. So I know there was a couple articles that came out about how in Europe they had like a few people that got Bell's palsy after taking the vaccine, and then there was guidance issued that if you have any like severe allergies, you should not take the vaccine, or at least, you know, your risk of some complication is higher. That's typical for basically every single vaccination. You know, some of those articles might have been alarming, but obviously you guys can read this and decide for yourself, you know, whether or not. So what I want to do next is jump down to this vaccine efficacy section. And here you will see this table right here. And you'll see this vaccine efficacy number of 95%. And then there's kind of this table of values here. And then they give some language here. But what I want to actually do is talk you guys through how they actually come up with this 95% number. And the reason I want to do that is because when I read this study, there's actually nothing in this document that shows or demonstrates how they actually come up with this 95% number. And so I was going through here kind of trying to divide numbers and seeing if I could get it 95%, and I just couldn't do it. So I decided to look it up, and I came to the CDC website here. You can look up vaccine efficacy or vaccine effectiveness, and they actually give you the equation here. So it says risk among unvaccinated group, subtract away the risk among the vaccinated group, and then you divide that by the risk among the unvaccinated group, or you just take 1 minus the risk ratio. So then the next question becomes, well, how do you calculate risk? So they have this example right here of calculating vaccine effectiveness, and they say data is from table 3.13, and basically you have to go to lesson 3 and then kind of click through some stuff, but you actually arrive at how they define risk ratios and stuff like that. So let's actually read this because it's kind of interesting. A risk ratio, also called relative risk, compares the risk of a health event, disease, injury, risk, factor, or death among one group with the risk among another group. It does so by dividing the risk incidence proportion, comma, attack rate in group 1 by the risk in group 2. The two groups are typically differentiated by such demographic factors as sex, for example, males versus females, or by exposure to a suspected risk factor, did or did not eat potato salad, or in this case, did or did not receive a vaccine. Often the group of primary interest is labeled the exposed group, and the comparison group is labeled the unexposed group. And then they actually give, it's kind of cool, they actually give an example right here of a vaccinated and unvaccinated scenario. So what I actually did was I took the numbers provided in this table and I put them in a little spreadsheet right here. So we've got this table right here should look identical to this table right here, which it basically does. We've got the vaccinated and unvaccinated. We've got those where varicella, in this case, their example is varicella. We're going to use COVID-19. And then we have not infected. So that's what we see here. Developed COVID-19, yes or no. Total, vaccinated, placebo, and then we'll calculate some risk factors here. So when you actually plug in these numbers, so the number of people who developed COVID-19 when they were vaccinated was 8. That's this number right here. The number of people who did not develop COVID-19 is actually this number, 17411 minus 8. Now you might say, Alex, why aren't you using this 18,198? Well, if you actually look at the footnotes here for A, this is the number of participants in the specified group. But if we look at what this number is here in parentheses, N2, this is the number of participants at risk for the endpoint. So what the endpoint is basically the endpoint of the study. And what you'll find if you look through this study more comprehensively is that there's a number of people who did not necessarily meet the endpoint. This is for a number of reasons. It could be because they didn't follow up with the second dose of the vaccine, or for whatever reason they decided to stop reporting after they received both doses or something like that. Basically just random issues with people's behavior that makes it so that the actual people who reached the endpoint of the study is less than the total number of people studied. So that's why we use this number in parentheses right here. So we've got 8. Here's that 17411. 17411 minus 8 is 17403. We have 162 in the placebo group right here. Similar thing, 17511, 17511 right over here. We've got some total columns. And then we calculate the risk in the vaccinated group. That's basically 8 divided by 17411. Get 0.00046 in terms of percentage. That's just given the second column for reference. Do the same thing for the placebo group, 0.925, and then we calculate the risk ratio, which is demonstrated right here. So here they have vaccinated divided by unvaccinated, 0.118 divided by 0.429. Similarly, we have 0.0046 divided by 0.00925, that's 0.05, comes out to 5%. And as you can see, you can take 1 minus the risk ratio, 95%, or you can simply take risk among unvaccinated group minus risk among vaccinated group. So that'd be 0.00925 minus 0.00046, and then divide that by the risk among the unvaccinated group, 0.00925, and you get this 95% number right here for that. So I thought that was kind of interesting. You can use this evaluation to, you can actually use this evaluation in a similar sense in the safety stuff as well. You can kind of gauge what the percentage chance is that someone will develop symptoms. But there is something a little bit subtle in this that is kind of important to note. You might ask yourself the question, well, don't these risk factors, aren't they dependent on the composition of the population? And at least if you didn't ask yourself that question, that's kind of the question I was asking myself. And so what's kind of interesting is how diverse they actually selected this safety evaluation to be. Now there's an upside and there's a downside to that. And let's just read for a second what kind of participants that they selected here. So here it says in phase 2 and 3, participants were enrolled with a stratification by age: younger adults 18 through 55 years of age, older adults over 55 years of age, and a goal of 40% enrollment in the older adult age group. Adolescents were added to the protocol based on a review of safety in younger adults enrolled in the ongoing study. So the age strata were revised as follows: 12 through 15 years of age, 16 through 54 years of age, and 55 years of age and older. The study population for phase 2 and 3 includes participants at higher risk for acquiring COVID-19 and at higher risk of severe COVID-19 disease, such as participants working in healthcare, participants with autoimmune disease, participants with chronic but stable conditions such as hypertension, asthma, diabetes, and infection with HIV, Hep B, or Hep C. Participants were randomized 1-to-1 to receive 2 doses of either the vaccine BNT162b2 or the placebo 21 days apart. The phase 2 portion of the study evaluated reactogenicity and immunogenicity, those are 2 kind of cool words, for 360 participants enrolled early on. So what's kind of interesting about this stratification is that it's very, very broad. And so you can imagine that if you have a population in this particular stratification by age, they also have a lot of people from different ethnicities and things like that. But you can imagine, like, if there are say cultural differences, and they also did different countries too. So if there's differences in sanitization practices or maybe healthcare between countries, or cultural differences, willingness to go to the doctor, willingness to get tested, things like that, then you might actually have a group that either has an extremely, extremely low risk rate, so that when you kind of average out all the risk rates, it might bias it to one side for one population anyway. Although I think that's probably why they do the randomized 1-to-1. Or what I was thinking is you might study a population that has a super high risk rate, say in your placebo group, but then you study a population that has like a relatively lower risk rate in your vaccine data group, and there might be a possibility that you just kind of underrepresent the risk rate in the vaccine group, simply just because of cultural differences or strange demographic differences or perhaps country of origin differences. That said, I think that's why they do the 1-to-1 thing, but that was kind of a question that I had. And so what I think would be really interesting is if, in engineering, we try to be as conservative as possible, meaning we try to plan for the worst, kind of hope for the best. And so it's curious to me how come they don't do like all the people who are going to get vaccinated have like they just pick everybody to have the highest risk demographic, and then they actually placebo the people who have like the lowest risk demographic. That way you're kind of getting a worst case on worst case for both. And so when you actually end up calculating the efficacy rate, it's like the best possible efficacy rate that it could ever be. But I'm sure that there's like a scientific issue with that proposal on my behalf. But still, I thought it was an interesting question. So with that, let's go ahead and just kind of round this out with looking at the safety summary. We already talked about the efficacy summary, which is a very kind of like rewarding result, 95% efficacy. I showed you guys how to calculate that. I raised some potential issues with that, but overall, it seems like, in my opinion at least, this is like a pretty sound study. But you guys can look at the study, or you guys can look at the data and the information and decide for yourselves You don't have to rely on the media or politicians or, whatever experts, quote unquote, you have in your life. You can read about this and decide for yourself whether or not you trust it. But let's go ahead and just round this out with a final safety summary statement. The information provided by the sponsor was adequate for review and to make conclusions about the safety of the vaccine in the context of the proposed indication and population for intended use under the emergency use authorization. The number of participants, again, was all these numbers that they have right here, meets the expectations in the FDA's guidance on the development and licensure of vaccines to prevent COVID-19 for efficacy and a median duration of at least 2 months follow-up after completion of the 2-dose primary vaccination series, meets the agency's expectations and FDA's guidance on its emergency use authorization for vaccines to prevent COVID-19. I think there was maybe a little bit of redundancy in that sentence, but I also may not have read it correctly. They go on to, again, explain kind of what the safety data showed, which is that it's pretty rare that you will have a extremely adverse reaction to the vaccine. And some people may have an adverse reaction, but it's not any higher than the adverse reaction rates in other populations, probably for other vaccines or for other things. So anyway, guys, we're going to wrap the video up here pretty soon, but I did want to share this with you guys. I'll put a link in the description below. Please go through this and check it out. And I hope that at least this gives you guys some food for thought or some rationale for yourself to at least consider as to whether or not you're going to get the vaccine. Personally, I probably will get the vaccine. And if I don't, my hope is that my parents will be prioritized because the biggest issue for me is not necessarily that I'm afraid of the virus, but obviously, there are people in my life who are a lot more worried about it than me, and I would like to be able to spend time around them and not have them feel like I am, risking their safety or whatever, just kind of being around. So all that being said, you can look at all the science and all the data and everything like that, and still, not. Let's say that the vaccine was 100% effective and the data showed that it was 100% effective for all time, forever and ever and ever. Does that mean that the government should mandate that people get vaccinated? Does that mean that the government should give out kind of social credit, like little, punch cards or whatever that show that you got vaccinated so that you can get into concerts and stuff? I think there's no amount of science that can ever make that moral decision. Now, my opinion on that is you should not force people to get vaccinated. It should be a personal choice. They should have access to the data. People are smart enough to read through this stuff and make their own decisions. Now, obviously, not everyone in the country can read, and even if you can read, you might not understand all this language, but the information is available to you. There are people around who can explain it to you, and as long as you're provided with the information, then you should be able to make your own decisions about whether or not you want to get the vaccine. At least that's my opinion. I will likely be getting the vaccine when it becomes available en masse to the general public. And that said, I think that the same approach needs to be taken for, lockdowns and closures and stuff like that. I think that people know what the risks are. Obviously, we've been dealing with coronavirus for the last year. People are aware of the science. People know where to go to look at the data. People can decide whether or not they want to look at the data. But I just think that, it's not really up to the government to decide for people what their risk factors are and what they should and should not do when it comes to something like this. I know that, we're not all out there intentionally trying to hurt people and cough on people and get people sick. I think it's pretty ridiculous that it just kind of seems like the government officials think that we're too stupid to be able to manage risk in our own lives. And, I do stupid stuff and other people do stupid stuff too, but we have the right to do that stupid stuff as long as we're not intentionally trying to hurt people with, nefarious will or anything like that. So anyway, I hope you guys enjoyed the video. Leave a comment below if you guys spot something in here that you think I missed that's interesting, or if you have some opinions on whether or not it's there's like a moral imperative to force people to get vaccinated just because of scientific data. I personally don't think that there's a moral argument there. I think that the individual should be able to decide as long as they're given the information. And I very much appreciate the fact that this information is available online. Thank you very much to all the companies working on the vaccine as well. You guys have done a lot of really good work. I think that this data is really compelling. It's a well-written summary, and there's not a whole lot of things that they missed in just this briefing document, which I'm sure is like a redacted version of, a giant report with pages of data. So anyway, guys, have a good night. Have a good rest of your weekend. And I will catch you guys in the next one. Later.